Nitrogen is an essential nutrient to plants. However, nitrogen fertilizers which increase crop yields also represent major cost in crop production. Also, increased use of nitrogen fertilizers affect global nitrogen cycle, depletion of ozone layer, and cause nitrate leaching problems in soil. Nitrogen use efficiency is relatively low in rice, as major part of nitrogen applied to it is released as nitrous oxide into the environment. Indian Council of Agricultural Research sponsored project, National Initiative on Climate Resilient Agriculture, NICRA, was initiated at Directorate of Rice Research with the aim of identifying and developing high nitrogen use efficient rice genotypes under low nitrogen to ensure low cost and environmental protection. Low nitrogen field was developed here without giving any nitrogen inputs for 4 years continuously. More than 800 rice genotypes were evaluated for more than 100 morphological, physiological and yield parameters under both low nitrogen and recommended nitrogen fields for 2 consecutive years. Potential donors for nitrogen use efficiency were identified and several mapping populations were developed using these donors and popular varieties for identifying genomic regions responsible for nitrogen use efficiency. These genotypes were also evaluated for heat tolerance in temperature gradient tunnels. In the present research paper, 78 aromatic rice genotypes were screened for physiological and yield traits under low nitrogen and recommended nitrogen fields during wet season 2011 and dry season 2012. Cluster analysis was performed for physiological and yield data and dendrograms were constructed. Based on these cluster analysis, genotypes were classified into high, medium and low NUE genotypes. Basmati 370 and Ranbir Basmati as high NUE and Kolajoha 3 and Ratnasundari as low NUE genotypes were selected for further biochemical and physiological evaluation. These selected 4 genotypes were evaluated for further 32 biochemical and physiological traits and 5 efficiencies related to nitrogen use efficiency: glutamine synthetase and nitrate reductase, chlorophyll content, carotenoids content, chlorophyll fluorescence, and photosynthetic parameters were measured in flag leaves. Grain weight, total dry matter, percent nitrogen content, and nitrogen uptake in both grain and straw were also measured, and 5 efficiencies related to NUE were calculated. GS activity in high NUE genotypes was 1.76 times that of low NUE under low nitrogen. So high NUE genotypes can utilize more ammonia as an alternate nitrogen source in nitrogen stress conditions. There was negative correlation between NR and GS activity, suggesting that GS activity was limited when the rate of nitrate reduction is more to manage the stronger flux of reduced nitrogen. Chlorophyll content was more in low NUE genotypes. Specific leaf area was more in high NUE genotypes, whereas leaf thickness was more in low NUE genotypes. There were significant differences in chlorophyll fluorescence parameters, but not in photochemical efficiency and electron transport rate. Low NUE genotypes exhibited significantly more photosynthetic rate under low nitrogen field. This observation indicates no relationship between photosynthetic characters and grain yield and also nitrogen use efficiency. Even though efficiency values related to NUE were more in high NUE genotypes, these values were not statistically significant. The reason for this may be that NUE related efficiencies are complex traits depending on more than one trait such as grain yield, straw yield, nutrients applied, nitrogen content of straw and grain, and nitrogen uptake by straw and grain. So the cumulative effect of all these traits might not give significant differences. The effect of different nitrogen levels on agronomical, biochemical and physiological processes in aromatic rice was studied. This study supported that Basmati 370 and Ranbir Basmati were more efficient in nitrogen utilization than Kolajoha 3 and Ratnasundari. These genotypes can be further utilized in breeding programs to develop high NUE varieties and also in molecular studies to identify genomic regions associated with nitrogen use efficiency.